Okay, let's just get back to where we left off. Sorry about this. I wasn't already booted up here for you. So we went through all the anatomy of contraction. We talked about the different types of fibers, the different types of contractions, and we left off with smooth muscle. So smooth muscle, again, we'll talk about smooth muscle more when we get into the digestive system more specifically. It's found in the walls of hollow organs. So here we're talking about blood vessels or veins and arteries, the digestive system like the stomach, small and large intestine, as well as the urinary system, and the uterus. Those contractions for childbirth are all brought to you by smooth muscle. They're also found within the skin attached to the hair follicles. Those erector pili muscles are a type of smooth muscle. The smooth muscle can actually stretch more than skeletal or cardiac muscle, which you may have known if you've ever overate at a meal, for instance, Thanksgiving dinner, right? You eat a big dinner and your stomach stretches a lot. That's all the smooth muscle allowing you to stretch like that. They also can maintain steady levels of contraction for long periods of time, moving that food through, for example, or staying tight in the case of high blood pressure. Smooth muscle anatomy, they're smaller in size than skeletal or cardiac muscles. They're more spindle-shaped as opposed to rectangular shape. They only have a single nucleus and no striations. The thick and thin filaments are not arranged in the sarcomere shape. They're kind of scattered throughout the inside of the smooth muscle cell. So it's a more random contraction. If you think about how smooth muscles contract, it's not just a shortening, it's more of a wave-like or blob-like contraction that the smooth muscles go through, rather than just a very regular and predictable shortening. There's no T-tubules. They do contain intermediate filaments and dense bodies that kind of act like a Z-disc, and those are within the cytoplasm attached to the cell membrane, and they don't have a well-developed sarcoplasmic reticulum. So their overall arrangement is very, very different than the skeletal muscle that we were just going through. Here you can see what I mean by their spindle shape. They are known as a single unit of muscle tissue. They're interconnected through special gap junctions, and again, they produce more of a wave-like contraction. Peristalsis is the movement of food through the digestive system, and that occurs in a very wave-like fashion, as those contractions are occurring more in waves than in just straight shortenings. There's no gap junctions in a multi-unit smooth muscle tissue. The cells can function more independently. This would be like blood movement through the arteries, rather. It's more of a specific pinpoint of control for the contraction, rather than the entire wave-like contraction that would occur in, say, the digestive system. As I mentioned, the contraction is very, very different. It's more blob-shaped or wave-shaped. They contract and relax involuntarily in response to the neurons of the autonomic nervous system, which we'll talk about later on. They can also respond to hormones as well as local events like changes in pH, carbon dioxide levels, temperature, as well as ion concentration. If you look at your two cells here, one is a relaxed cell, or one is a relaxed cell, and one is a contracted cell, and you can again see it does shorten, but it also fattens. It has a very different type of contraction than our skeletal muscles do. Cardiac muscle is the type of muscle that composes the heart. It's kind of an intermediate structure between our skeletal muscle and smooth muscle. It's a little more neatly arranged than our smooth muscle is, but quite different from our skeletal muscle. It's larger than smooth muscle. There's cylindrical myofibrils, there's sarcomeres, there are T-tubules and sarcoplasmic reticulum. So in that respect, it's very similar to our skeletal muscle. But they communicate through gap junctions called intercalated discs, which are going to attach adjoining cardiocytes or cardiac cells. And these cells are branched, whereas our skeletal muscle cells are not branched, nor are the smooth muscle cells. Here you can see those special connections in, or rather the branching that we're looking at here, branching of our two types of cells. You can see the T-tubules kind of invaginating into the cells themselves, and you can see these special intercalated discs, these wiggly lines connecting the cells together. Cardiac cells Some of them are special pacemaker cells. They're specialized heart muscles. They are under autonomic nervous system control, and they can either speed up or slow down the contraction rate. And then there's the cardiac cells themselves, which are going to respond to the pacemaker cells. So pacemaker cells literally set the pace for your heartbeat and are kind of like the conductors of your heart contractions. The cardiac cells are autorhythmic, meaning they can contract on their own without the nervous system. In other words, they just respond to the pacemaker cells. They produce a twitch about every 70 to 80 times per minute, which is why our heartbeat is generally around 70 beats per minute. Skeletal muscles work in groups, as we know. They're attached to bones via tendons. Two bones are either involved in a movement of one joint or stationary, and the other can be movable. The one end is called the origin, the other end is called the insertion, and the middle part is called the belly. The end that attaches to the stationary bone is the origin. The end that attaches to the movable bone, usually via a tendon, is called the insertion. So let's take a look at these. For example, our biceps brachii. It's called biceps because there's actually two heads to our bicep muscle. See here how it kind of splits off into two? Well, the origin is going to be up here closer to the midline of the body. Typically the insertion is the distal end of the muscle. So in this case down here on the radius bone is our insertion for our biceps brachii muscle. Muscles work together in antagonistic pairs. If you've ever done any type of muscle training, you probably know this. If you work your biceps, you also have to work your triceps, right? The prime mover is also called the agonist. This is the muscle that does the desired action. So if the desired action is flexing the elbow, the main prime mover in this case is going to be that biceps brachii muscle. The antagonist is the muscle with the opposing action. So if our prime mover or agonist is the biceps brachii, our antagonist is the triceps brachii. The synergist is going to help the agonist function more efficiently. So it's going to help hold something in place. It's going to assist with the overall movement. If we're talking specifically about the biceps brachii, there's another muscle that is associated with flexing the elbow, and that is called the brachialis muscle. It sits just deep to or underneath of the biceps brachii. So the synergist to the biceps brachii would be the brachialis muscle. Fixators are those that stabilize one of the bones so that the agonist can move the other bone more efficiently. So an example of this would be the scapula. Here are some major skeletal muscles. In lab, you're going to be going through some specific skeletal muscles. Your lab instructor may also have you know the origins, insertions, and particular movements of some of these muscles. Here are the muscles of the posterior. I'm not going to lecture on them. You may see some of the major superficial muscles on a test or quiz, but I won't test you on the specific small muscles. So any of the muscles that you see here on this slide or the previous slide would be fair game for a test or quiz in terms of their movements, origins, or insertions, but you don't need to know all of the smaller muscles specifically for a test or quiz. That's up to your lab instructor. I'll stop the lecture.